about the carbohydrate metabolism. Actually, carbohydrate metabolism is a series of the lectures, and you have lipid metabolism, you have protein. I will give you the carbohydrate part. I think it will be 3 to 4 lectures. We will start with the first one. Try to listen, try to concentrate from the first lecture, because it will be continuous. If you don't understand the first lecture, you will not understand the second and third. Okay. Okay, so first slide is talking about the beta. Please respect the teacher, please respect yourself and the whole, okay? Alright, so first of all is the overview of metabolism. Is generally speaking, it's composed of the major source of the energy, which is, we know, is a carbohydrate, is a protein and fat. We will have this part together. We will concentrate on the carbohydrate metabolism, and we will return back to this slide at the end of our lecture. So we will start first with the stage 1. We will talk about carbohydrate metabolism until it becomes glucose, and how glucose become another molecule, and going through all the cycle finally to generate ATP. So the first lecture will talk about the glucose, or first of all, carbohydrate in general. Carbohydrates are molecules consisting of carbon, hydrogen, and oxygen. Okay. Carbohydrates are the largest source of dietary calories for most of the world population. We know that carbohydrate is our major ingredient in our food, in our life, so we depend mainly on the carbohydrate for the energy. It has a diverse function as energy storage. We can store it in the form of glycogen, or in a form of structure formation like cellulose and chitin. This is mostly the plants. How the carbohydrate classified? Anyone has any idea? Or structurally, we can say. Okay. And the other one, yes? Good. And? Polysaccharide. Okay, good, very good. Now we have carbohydrate is classified into monosaccharide, like example, glucose and fructose, that have a single carbon chain. And we have a disaccharide that's composed of lactose and sucrose with 2 carbon chains. What's the ingredient of the lactose? Composed of which? It's 2 carbon chain. Composed of
galactose and
glucose. What about sucrose? Composed of glucose and
fructose.
Fructose, yes. So sucrose composed of the fructose and glucose, and lactose composed of galactose and glucose. So you have glucose in each. And we have oligosaccharides. Sometimes they will classify it also as a polysaccharide, or sometimes they will divide it into oligosaccharide, which has a 3 to 10 carbon chain, like dextrine. You will see what's dextrine in the next slide. And we have polysaccharide with more than 10 carbon chain, like a starch, which is a complex sugar, multiple glucose connected to one another. It's called a starch. So this is just an example. You don't need to memorize this, just to see. This is a monosaccharide composed of single chain, and it too is connected to one another. Okay. We will start with the carbohydrate digestion and absorption. First of all, anyone has any idea where the digestion starts? Yes. By effect of which enzyme? Yes.
Amylase?
Yes? Yes, it's called a salivary amylase. Very good. How it will be, we start at the mouth, okay? How it will be stimulated, how it works, how this enzyme will be secreted. Yes?
When the chemoreceptors in our body.
Yes, by which mechanism? Yes? It's simple. When you start mastication or chewing, proceed chewing this okay? Salivary amylase, this version. So the digestion of the carbohydrates starts in mouth. Start in oral cavity by action of salivary amylase, and continue in duodenum by pancreatic amylase, and complete in duodenum and jejunum by maltase, lactase, isomaltase, and others. The entire fiber cannot be digested by human enzyme. Intestinal tract. We don't have an enzyme to start breaking down fiber. That's why we pass fiber and digest it. So this is how we'll start. When we eat high carbohydrate meal, what's the main component of the high carbohydrate meal? Is it glucose? Is it glucose? What's the main component of the carbohydrate in our food? Yes? Free glucose? So why we digest it if it is free glucose? Yes?
Sucrose, then break it down to
Main component. What's the main component? Yes.
that
Yes, starch. What's starch?
polysaccharide.
Yes, it's a polysaccharide. Okay, thank you. It's a polysaccharide. It's composed of multiple glucose connected to one another. It's glucose alpha 1,4, glucose alpha 1,4, sometimes glucose alpha 1,6 glucose. It's the complex sugar. So most of the carbohydrate is in a form of starch in our food. We have rarely or very tiny amount of free glucose in our food in a form of carbohydrate. That's why we have to digest it. And we have also sucrose, some amount of lactose in the milk and other dairy products. So we start with the salivary amylase. We said we start chewing. The chewing will activate salivary amylase. And salivary amylase will do what? Will do breaking down of the starch randomly. It will randomly break the starch. What will generate? It generates oligosaccharide. We said it's from 3 to 10 carbon chain is called oligosaccharide. Okay, this is called dextrin. And then in the stomach, is there anything happening in the stomach regarding carbohydrate metabolism? Any idea? Any idea what happened to carbohydrate metabolism or digestion, you can say, in the stomach? Any process will happen? Yes or no? Yes?
It will be catabolized to glucose.
No. In the stomach, actually, there will be nothing or no digestion for the carbohydrate, okay? So in fact, the acidity of the stomach will kill salivary amylase because salivary amylase cannot work in acid medium, okay? So in the stomach, nothing will happen. Then dextrin will go to where? To small intestine. It will see another enzyme that's called amylase, but this is a pancreatic amylase, secreted from the pancreas. Actually, it finishes the job of the salivary amylase. It further breaks the starch, okay? Or we can say further break the dextrin. So it will break dextrin into what? Into disaccharides, into maltose, isomaltose, sucrose, and lactose, okay? So we have first polysaccharide in the mouth, then broken down to dextrin, nothing happening in the stomach, then go to the small intestine. By pancreatic amylase, it becomes disaccharides, okay? What's maltose? What's maltose? Yes? Yes, glucose and glucose. What about isomaltose? What's the difference?
It's basically the same thing, but in a different shape.
How? Yes. How? Yes, very good. Yeah. Actually, maltose is also disaccharide. It's glucose alpha 1,4 glucose. What's that? It means that a carbon chain or carbon number 1 in a first carbon chain attached to the carbon number 4 in a second carbon chain. This is meaning of glucose alpha 1,4. 1 is attached to the fourth, okay? While isomaltose is glucose alpha 1,6 glucose. So it's attaching the first carbon of the first chain to the sixth carbon of the second chain, okay? So it's alpha 1,4, and this one is alpha 1,6. As we know, the sucrose and yes.
What's the difference in function between them? Why one of them?
Yeah, because we need different enzymes in order to break them down. Okay, we cannot absorb disaccharide. We have to break it into monosaccharide in order to be absorbed in the intestine. Okay, so unlike proteins can be absorbed in a dipeptide form, while the glucose, or we can say carbohydrate, cannot be absorbed in a di form. It should be further break down into mono form. So it will see the third enzyme that's called brush border disaccharidase. It will be secreted from brush border of the small intestine, and it will generate monosaccharides, mainly glucose and a smaller amount of galactose and fructose.
هذا منيح.
كان يا؟
نفس كده فقيت يعني بس
Okay, so what are these disaccharidases? Our maltose will be break down by an enzyme called maltase. And say the ASE, like what I have shared, this is an enzyme. So maltase will break down its target, which is maltose, into 2-glucose. Isomaltase will break down by isomaltose will be broken down by isomaltase into 2-glucose. And sucrose will be by sucrase into fructose and glucose, lactose by lactase into glucose and galactose. All these enzymes are called brush border disaccharidase. Okay? According to the name of the target, it will be changed. Then we will continue with glucose. So now we have glucose ready to be absorbed. Now we have to know little about the absorption of the glucose. We have an importer or transporter. This is called a sodium glucose transporter, meaning that the glucose will be transferred from the small intestine into the cells of the small intestine by the help of the sodium. Okay? Sodium is present inside the cells of the small intestine. They will come down to the lumen, and glucose go up from the lumen to the cell. Okay? So they will be exchanged. Sodium will come down to the lumen, while glucose that's present in the lumen will go up into the intestinal mucosal cell. This is called a sodium glucose supporters. So they will help each other. This will come down and glucose will go up in order to enter into the circulation. So here is from intestine going to the bloodstream. Okay, so now we know the digestion. We have to break the complex carbohydrate into monosaccharide in order to be absorbed, because we cannot digest oligo or polysaccharide in our intestine. It has to be break down into the glucose. How it will be absorbed? We said by help of the sodium, it will go into intestinal mucosal cells. And then how from intestinal mucosal cells enter into circulation, enter into all blood. So it's carried out from the gut to absorb the cell by active transport. This is called a sodium glucose transport. Then, from absorbed cells to other cells by facilitated diffusion, which needs glucose transporter. طبعا كم جربت شفنا السيل كان intestinal وتبع healthy sodium. دعاء له passive diffusion شفنا circulation. بعشان شو circulation إن جاليرو إيش كده سبيعة. شلون شفنا organ كان معنا. شلون شفنا liver, شلون شفنا brain. Because we know every organ in our body has glycolysis. Every living cell needs glucose in order to live. Okay, including our cell. So, بهلبي مجموعية transporter بيانا تجي glucose transporter, abbreviated GLUT. So it starts from 1 to 5. We have GLUT 1, 2, 3, 4, 5. So these glucose transporters are, as I said, it's abbreviated GLUT. It's called GLUT 1. That's present in most of the tissues like the brain and red cells. It has a basal uptake of glucose. ما نحكي basal uptake of glucose. يعني بردوان لريئا GLUT 1 glucose جتنا أو سلانة وكو chain وكو brain وكو red blood cell. يعني even أجابيت بيريدي hypoglycemia still a big glucose بردوان جتنا أو سلانة. Okay? This glucose يعني أونا affinity هي بو glucose حتى أجا glucose لوش بين بردوان إبا بخوي بناو brain وبناو RBC. This is GLUT 1. Okay? While GLUT 2, which is present in liver and pancreatic beta cells, it acts differently. In liver, it will help uptake and release of the glucose by liver cells. While in the pancreas, It will help as a beta glucose sensor. Because we all know that, what's the function of pancreas? Yes. Yes. It releases insulin, but this is what I need. Yes.
Helps digestion in intestine.
Yes, very good, very good. But this is, yes?
Releasing enzymes.
Yes, like pancreatic amylase, as we saw it, and many others. So, the GLUT2 in pancreatic beta cells had different function. It will act as a beta glucose sensor. It will sense the high level glucose in our blood. So when it senses high level blood glucose, it will start secreting insulin in order to regulate that high level blood glucose. Okay? كبلد glucose برزة بيتوا أم GLUT ولا سر كان شو هي بنكرياس هي سنسي أكاد بالمقابل شيء أرجع طبعا مجموعين ميكانيزم زورة كشال إنسولين أرجع بالمقابل إنسولين أرجع وأويكا ريجوليشني همو أو جلوكوز كان نوع جسمي توعه وينبت hyperglycemia ينبت diabetes. Okay? And we have GLUT3 also is present in most of tissues. تقريبا همان شيء GLUT1 أكا. It has a basal uptake. GLUT4 is present in skeletal muscle and adipose tissue like fat cells. Adipose tissue means fat tissues or fat cells. Insulin stimulated glucose uptake and stimulated by exercise in skeletal muscle. طبعا GLUT4 effect بس بالإنسولين إيش أجر الإنسولين هبل لما خلينا نتوقع الإنسولين هبل أو كات stimulate شيء كات كواي كان جلوكوز تناول مصلك كمان يعني لحالتي exercise stimulation أكا GLUT4 يتسر wally cell مصلك كان وش جلوكوز كات تناول مصل cell. Okay, so GLUT4 we stimulated by two things. First, insulin number 1 and exercising muscles. Okay. تركز إشكالي هي تركزتك. GLUT4 مع الأسر muscle cell هي لقل adipose tissue. Okay. بدوش stimulation ده insulin. أجر insulin هبل لبه stimulation أكا يعني وايد أكا شيء به glucose muscle كودي بكري يعني glucose تناول muscle cell كان. Okay. يعني exercising muscle لحالتي exercise يشاب GLUT4 يتسر cell membrane أكا glucose بعمخه. وش شيء بوي كمسل كا بكاري بينوكو energy. So it will be stimulated mainly by insulin and exercising muscles. دواء أو بسرعة نحب نقول أخير حاضر عشان بوي كات كمان هذا كلا يخوت عنا تكان بلوس نشيتي أنا جيش دواء بنجات. Now we will start with the first carbohydrate metabolism, glycolysis. I think most of you hear about glycolysis. What's glycolysis? What's glycolysis? Well,
The process of breaking down glycogen to glucose.
Yes.
The process of breaking down glucose into ATP.
Yes, good. And what's the final product of glycolysis? Yes. No, glycogen is a storage form of glucose. Okay. Yes. Yes, ATP. And the molecule that generates is? Yes, pyruvic acid and pyruvate. Yes. So glucose is 6 carbon. It will finally generate a 2 pyruvic acid or 2 pyruvate, which is a 2,3 carbon. So there will be no carbon loss in glycolysis. Glycolysis it happens in cytoplasm of the cell because we have a pathway that happens in the mitochondria. It consists of 10 reactions. Okay, so we have 10 reactions in glycolysis. 3 of them are irreversible, which is the 3 important steps in glycolysis. You have to know it. Which is the first one, 3rd, and the 10th reaction. It is 10 steps. You don't need to memorize all 10 steps, okay? Just 3 steps are very important, which is our 3 major enzymes in glycolysis. What happens, as you said, glucose converts into 2 pyruvate. 6 carbon convert to 2,3 carbon pyruvic acid or pyruvate. What's the aim is the release of energy in the form of ATP. And what happens to the pyruvate? If the cell has mitochondria and oxygen, glycolysis continues aerobically. If either mitochondria or oxygen is lacking, glycolysis may occur anaerobically, such as in erythrocyte and exercising muscle cell. We will show you. I will show you on the diagram better how it works. So this is the glycolysis. It looks complicated, but just as I told you, it's a 3-step you have to know. I will mention all 10 steps, but the most important one, I will tell you what you have to know, because these are the 3 key enzymes in the glycolysis. By the way, when you graduate in your lifetime as a physician, you will never see a patient with the enzyme defect in glycolysis. A partial enzyme defect do exist in glycolysis, but a complete enzyme defect is incompatible with life. يعني هيش كسانية بتواني complete enzyme defect يهبط لglycolysis بتواني بشي. So it's incompatible with life. Partial enzyme defect here, لأني يعني كباشي نبت، بلام it do exist. But the complete enzyme defect in any steps of the glycolysis is incompatible with life. So, glycolysis happens in every cell in the body, happens in every organism in the body. It starts with the glucose, as we know, and ends with the pyruvate. So the first step in glycolysis, once the glucose enters into the cell, this whole is the cytoplasm of the cell, okay? It will see the first important enzyme and the first important step in the glycolysis that's called a hexokinase. هكاتي إيوا أم كاينيزة تنبيني لكوتاي إنزيم كيا بيتا بيتا كاينيزة شيكات فوسفور أخذ سر تارجتك target hexokinase شيلera glucose. Okay, so whenever we have hexokinase, mean it will put a phosphate on the glucose to generate a glucose 6 phosphate. Now ده هالكاينيز اللي صار إنزيم ده بمعنى هو phosphate أخذ سر targetك يعني أخذ سر substrate. So here, the hexokinase will generate glucose 6-phosphate out of the glucose. It will use phosphate شو بكارينه؟ phosphate ATP بكارينه. ATP أقربه ADP. ليه برسالة بلوسبب؟ هدف في glycolysis شيء generation ATP هم متعنزان. So at the beginning we lose ATP. We use ATP. Why? Why we generate glucose 6-phosphate? Yes.
It's to activate the glucose. It makes it an anion and it also activates it, so it becomes more reactive for metabolic pathways.
Yeah, that's right. But the main purpose.
Why
why generate glucose 6-phosphate? Why not glucose? Why we use ATP when we need it? Yes? Yes, very good. The main aim is to trap the glucose inside the cell, okay? Because the glucose, if it is present as a glucose or it's remained as a glucose, it will get out from the cell by effect of GLUT, as we talked about. But when you put a phosphate on glucose, it will be trapped inside the cell. So it will be going forward for further metabolism. So this is the main aim, in addition to what your colleague says, become active, become anionic, but the main aim is to prevent glucose 6-phosphate or glucose to go out from the cell. So hexokinase, present in every cell in the body, is a very important enzyme. It will generate glucose 6-phosphate by using ATP and convert it to ADP. In addition, we have another enzyme here, hexokinase wire, glucokinase. أما نكراهه لليفراهه mainly, okay? باش كي لبنكرياتيك بيتا سيلا كان يشاهد بلان mainly كوب ميتابوليزم نكراهه لنا ليفراهه. بوشي شكل دواتر باس أكلي باسي أكين كا لليفرا تشيرو بوشي لليفرا جلوكوز ميتابوليزم جوازه لبرو يعمل glucokinase معناه. By the way, أم hexokinase أجا glucose 6-phosphate زور به automatically تشي بس shut down ببرد. hexokinase shut down okay? يعني شون كالتا inhibition ببرد glucose 6-phosphate will inhibit the hexokinase if it is too much. يعني كبرودكتك زور به hexokinase شي بس shut down ببرد. بالله أملاً كاتا كيرتل يشكردن glucokinase يشكل فاوقع. فglucokinase ببرودكتك inhibit نابت ببرودكتك كم نابت. بوشي شو hormonal regulation هي. هرمونات شيء insulin. So كinsulin أرجع stimulate شيء glucokinase كا. لبروا كا glucose metabolism the liver will continue while in other cell will stops. Okay? تيجيش نيابي لمو. سيريكا hexokinase ده مو سلا كان بلى. باشا سرچا. hexokinase يكم إنزيم مهمة للglycolysis. باشا it is irreversible. جوب قرقسمة كمان ما تبي عشان سبب. Hexokinase. This is the first important enzyme in glycolysis. Okay? It's present in every cell. Which man هركاتي enzyme kinase بيو بتشيكات phosphate أباد إخذ سر تارجتك. يعني تارجتك اللي ركيا glucose إكابة شيء glucose 6-phosphate. باشا بلام أجا glucose 6-phosphate زور بو شيء كايا inhibit enzyme كي خويا هذا بس لو زيادة من دروز مكان. أوكي؟ كهذا شيء كا inhibit hexokinase كا. لو كانت شيء بيستمر دوام من إيشا glucokinase. بوشي شو glucokinase ببرودكتك inhibit نابه. شون inhibit يعني شون stimulate يعني شون inhibit hormonally. شو stimulate كا insulin. ماشا كواتا that's why glucose metabolism للسيلا كان كبيريست يعني للسيلا كان يتر جسم معنا أوستين يعني glycolysis أوستين بلام لليفرا it will continue by effect of glucokinase. Okay? Is it clear? Okay. So this is the first important step, as I told you, and then we have a glucose 6-phosphate will convert into fructose 6-phosphate. So it's not important. And then we have a fructose 6-phosphate will convert by the most important, perhaps the most important than all of others, is called a phosphofructokinase 1. It's abbreviated PFK1, and it's PFK1. Basha? Phosphofructokinase 1 شيكا سيري كان جاية الكاينيز معنى كواتا مش شيكا فوسفوريك يترخى تسال fructose 6-phosphate باشا ببقارناني ATP شي درسكن fructose 1,6 bisphosphate يعني ديرا fructose معنى 2-phosphate باشا so PFK1 شيكا fructose 6-phosphate المربع fructose 1,6 bisphosphate ببقارناني ATP جاري كتر because it is a kinase enzyme similar to hexo and glucokinase. Okay? بوشيم enzyme مهمة لبروي كريجوليشن كي زور مهمة. شلون control أكري. This is the key enzyme in the glycolysis. This is the enzyme that tells the cell to continue glycolysis or stop. باشا يعني أما كليلي glycolysis أما بي الله بوستا يعني بردوامة. Okay? بوشي لبروي كان بتشي inhibitor بيا بتشي stimulator برا AMP. Stimulate يكا inhibit يكا. أكيد شو قصاني ده صوب ربي زحمة؟ ده. حاضر كان طلع. Yes. AMP شيء يكا. شو أنت معنا؟ Positive. Yes. So positive effect يعني activate يكا. How? Why? Okay, so AMP is indication of what? Yes, very good. So whenever the AMP in our cells rising, meaning you are exercising too much, you are losing energy, so you need to make more energy by activating a key of glycolysis, which is a PFK1. Okay? كواتبنا كتو exercise كي سيركي exercise كي خوتي لا أكيد وردور ده ATP كان هم يصفبت شي برزبته وبرادر كتانتي AMP برزبته كي AMP برزبو أنا من أمريت إنرجي زيادة دروس كم يعمل بوست glycolysis من إنرجي دروس كم بوشي ويبتوان بردام من الرياضة كم ويبتوان activity كم بردام so AMP يتشي أكا activating clearly glycolysis أكا كيا ك PFK1 okay so أجابي لكي تو بيزعل على بريشكي شكا أجابي لكي تو لجسمي خود ولا بيشو بهني برجان برجاني خود أتواني توقعي كي كبوتشي AMP stimulating PFK1 okay. What about ATP? Of course, it will do the reverse when you have a lot of ATP and you do not exercise a lot or you don't have activity. So you tell PFK1, stop glycolysis. Okay? Shut down glycolysis because we don't need further ATP to be produced. Citrate, I will not tell you about the citrate. In next lecture, we will explain how the citrate will turn off the PFK1. So this is the second important step. We said why it's important because of its regulation by AMP and ATP and citrate. Now we have fructose 1,6 bisphosphate split into two products. This is dihydroxyacetone phosphate and this is a glyceraldehyde 3-phosphate, okay? This is not important how it generates, by which enzyme it generates, and how it works. Okay, so the glyceraldehyde 3-phosphate or dihydroxyacetone, this is a reversible step. You see if there is a 2 arrow on the head, again, we need to have a reversible step, يعني أتواني interchangeable. أتواني dihydroxyacetone phosphate, glyceraldehyde and forward. بيدن بن. هي متن لبيداتو، هر كسي بقصة كردن هاتو بيزنا كبدن حاضرتو. Now we have glyceraldehyde 3-phosphate. أجر dihydroxyacetone phosphate بردام نبي أتوانا بقرته بglyceraldehyde 3-phosphate. كواتن ليرة وهموشتن it will be multiplied by 2. همي multiply by 2 بأجر 2 molecule glyceraldehyde 3-phosphate معناه, okay? So glyceroldehyde 3-phosphate will convert into 1,3-bisphosphoglycerate. This step and the enzyme is not important, but just know that this is the only oxidation step of the glycolysis in which a molecule called NADH will be generated. طبعا دواتر أزعن NADH شيء وشون بكاريا وشي دروز أكا. So just know this is the only oxidation step and it will generate NADH in glycolysis. That's enough. About 1,3-bisphosphoglycerate will convert to 3-phosphoglycerate. Again, the name of the enzyme is not important. But just, you have to know that this step is called a substrate level phosphorylation. Anyone has any idea, according to the name, tell me what's substrate level phosphorylation? Yes? No. Yes? Any idea? It's okay, it's wrong. Yes? How? From where? Yes? Yes, very good. So this is meaning that a substrate is used to generate ATP, okay? So the phosphate from this substrate, which is 1,3-bisphosphoglycerate, will convert ATP back to ATP. So from this step, the first ATP has been formed in glycolysis. This is the first of 3 examples in our body. This is called substrate level phosphorylation. Then has the example of substrate level phosphorylation. Duanen the glycolysis. Kata substrate level phosphorylation shibu substrate ka khoy. Ya target ka khoy. Phosphate ka ya ba ADP u ATP ma bodu Okay? This is the meaning of substrate level phosphorylation. Now, 3-phosphoglycerate will go to 2-phosphoglycerate and finally go to the PEP, which is called a phosphoenol pyruvate. Phosphoenol pyruvate is the highest energy compound of the cell. Okay? It's very high energy compound, even more higher than ATP and any others. So it will see the third and last important enzyme in glycolysis, which is called a pyruvate kinase. Okay? Again, this is the second example of substrate level phosphorylation. Shibu substrate level phosphorylation, Digestion. Bale, Shibu. Okay. Substrate again? Okay, good. Substrate level phosphorylation يعني substrate aga خوي. Substrate aga كامل. Substrate aga ويا كلا بيش enzyme كان أما بيه just substrate. يعني أم substrate ده فوسفاتي ليه ولا كده أخذ السر ADP بويكا ATP دوزكا. Okay, so the substrate itself is used to generate ATP out of the ADP. Okay, Digestion. Okay, so this is the second example. The pyruvate kinase step is the second example of the substrate level phosphorylation, in which the energy of PEP here is used to generate ATP out of ADP. يعني فوسفات هي أميات أشوف السر ATP كو شيء دوسكا ATP دوسكا. لو أنا برساكي بغيت ستيبي أخير بود شيء مهمة. طبعا ليرة regulation كي مهمة أبي شوكو PFK1 بلا ما يكون مهمة ليرة أوياك there's a disease related to the PK deficiency. We said أجر complete enzyme defect تهبل glycolysis is incompatible with life. بلا partial enzyme defect معناته هو كل ليرة. كبي أو pyruvate kinase deficiency. It's a partial enzyme defect. It's not complete enzyme defect. So what will happen if the pyruvate kinase is deficient? So PEP will generate in pyruvate, which is the last product of the glycolysis. I will talk about pyruvate kinase, but let me finish the pyruvate matter. So once the 2-pyruvate has been generated, if we have oxygen, it will enter the mitochondria and continue aerobically. The TCA cycle. Again, oxygen nabet, وكوشة. Just tell like oxygen minia, but anaerobically glycolysis برداوم ده. كيف No. Red blood cells and
Brain.
yes, I hear something. Yes, exercising muscles. Yes. Can take exercise أو oxygenate كناسي بأي هشتا بشي أو exercise أو مصلحة كبردان contraction أكا. So بشي كيف بتشي يبقى anaerobic glycolysis. So the pyruvate will continue by enzyme those called lactate dehydrogenase form a lactate. This is an anaerobic process. Okay, back to the pyruvate kinase. تا السكت ممكن إشكالك here. Okay. We said why the pyruvate enzyme is important because there's a disease related to the pyruvate kinase deficiency. It is the second most common cause of hemolytic anemia. What's hemolytic anemia? Yes.
Destruction of the red blood cells.
Yes. If we have a second most common cause, so perhaps we have a first or the most common cause. Any idea what's the most common cause of hemolytic anemia? The second position has been taken by PP deficiency. What's the most common cause?
Iron deficiency.
No. Hemolytic anemia. Yes? No. Yes? Oh, deficiency. Any idea? Yes? No. Yes? It's a hemoglobin deficiency. Hemolytic anemia develop hemoglobin deficiency. But what's the cause? The most common cause of hemolytic anemia is G6PD deficiency. G6PD أول نخوشي كناومنا وشتازي عترباو كألن أما نخوشي تشي نخوشي باقلبو بوشيش كباقلي خواردوا خوني خون بودو نتعم بيستولا كس. Okay, so this is the most common cause of hemolytic anemia. It's called a G6PD deficiency. Okay? The second most common, as we said, is a person in the glycolysis called a PK deficiency. So what will happen in PK deficiency? There will be hemolytic anemia, there will be increased 2,3-BPG. What's 2,3-BPG? I will tell you. And there will be this important part, it's called no Heinz bodies. Heinz body اللي هي the G6PD deficiency. أما ماركي G6PD deficiency. Again, Heinz body بين. طبعا Heinz body ده جاء مايكروسكوب عامل لنا نورة بلا سيل كان. أو لا بلا. نقلتهم بلى. لجا مايكروسكوب نورة بلا سيل كان أمير ريف Heinz body تشيل destruction hemoglobin لنا نورة بلا سيل كان. توبل أبدا عليها. لجا مايكروسكوب عامل يوم دوتة بشان ثانية ب G6PD deficiency كباسمك. Livia marquee G6PD deficiency is presence of the Heinz bodies. But the PK PK has no Heinz body. Okay, so this is the main difference in these two. Both develop hemolytic anemia, but there will be no Heinz body in a PK deficiency. What's 2,3-BPG? Agar seni erakin, agar pyruvate kinase deficiency habit, chi royat, there agar bashakum deficiency habit, chi kobetawa, era kobetawa. Why? يعني agar min إيه إنزيم كم defective ne إنزيم كم بجواني إيش نكات أتواني هم PEP بقرا pyruvate نخير كاتب بشكي أما كوبيت باشا أم step-up reversible سرك بدو شيء زابه 2 phosphoglycerate listen carefully and then go back to 3 phosphoglycerate back to 1,3 bisphosphoglycerate and in RBC we have another step called 2,3-BPG or 2,3 bisphosphoglycerate will be accumulating in our red blood cells. Okay, again, so when we have defect here, imagine we have an X over pyruvate kinase partially, PEP will accumulate back to 2 phosphoglycerate, back to 3, and 1,3 bisphosphoglycerate, and this step in RBC called 2,3 bisphosphoglycerate will accumulate. So that's why we have increased 2,3 bisphosphoglycerate, just want this one, or 2,3-BPG. Okay? Isfadi chia both shibasha is a NAD or NAD, the only oxidation step. Okay? Fa'era basha, tegishtu? Okay. Okay. Alkas persari habu, atwane about persari. Yeah, done. Please don't let me remind you every time. Be quiet. So, as we said, sorry, this is action. All right. Now, we said, this is again similar to the figure, insulin-induced uptake of the glucose in several tissue, in particular muscle and deposition كباسمان كده وده تفوتان خانم ربجني كده ولا جل slide يرسمك يا. طبعا in the red blood cell, the first site of ATP formation may be bypassed. ما بس يوهيك هنجار 2,3-BPG through سبين. As we said, 1,3-bisphosphoglycerol is converted to 2,3-BPG, can bind to hemoglobin, decrease its affinity for oxygen, and make it available to tissue in some circumstances for healthy areas. طبعا خوي طبيعي 2,3-BPG لناو هيموغلوبينا هيا لناو رد بلاسيلكانا هيا بوشي بو اي كاتيكا رد بلاسيلكي تشو بو من اكسجين هلا اجري اجاتي اكستريميتي كان بتواني اكسجين كا بتيشو بيتو كانت 2,3-BPG تشي كان ستيموليتي اكسجين اللودين اكا يعني هو اكا اكسجين كا هيموغلوبين كا ببيتو باشا طبيعي اما نورمال باسكين 2,3-BPG شكيتشي للرد بلاسيل stimulate oxygen and loading يعني واكا اكسجين كله هيموغلوبين كبيتو بيا بخانه كانيتو باشا كواتا decrease oxygen affinity and stimulate oxygen and loading طبعا كاجر too much 2,3-BPG وهبت بكل حالتي PK deficiency لا بحالتها هيموغلوبين ناتواني برا بيجي اكسجين هول بقري شو هيج affinity هي اكسجين نماو ك 2,3-BPG كي زور هنا ورد بلاسلكان لبرو زور زو هيموغلوبين اكسجين كجا بدنا اصلا ناتواني باين جيها ويشتي كي باش نيه ك 2,3-BPG زياده ابي هبت بلام الليفلي خوان اوكي So this is about the control of glycolysis. Before going to that, any idea what are the types of the metabolism that happens in the RBCs? RBC do metabolism متاعها. شو إيش بريطان هنا؟ ليش كمان باسكت شيبو? Glycolysis, yes. And the second one I will tell you is hexose monophosphation. معناه ويا no lipid metabolism, no protein metabolism will not happen inside the RBCs. كواتا بوشي كبيكي deficiency ما نبي سلكاني تر effect نابت خو glycolysis نحن مش هنكار ويا بوشي بس RBC بهيموليتيك أنيميا بوشي هيموليتيك أنيميا ويا كبيكي deficiency ما نبي بوشي سلكاني liver destruction نابي بو سيلي مسلكان destruction نابي بو بس RBC yes آخر لبروي كتاقة سورسي ATP وتاقة سورسي إنرجي وRBC شيء glycolysis لبروي كdeficiency كمل الإنزيم glycolysis يعني استبيكي glycolysis يكسر شيء affectable RBC يكسر بالdestruction hemolysis okay وين على hemolytic anemia this is the major sign of peaky deficiency لبروي liver cell كان muscle cell كان أتوان triglyceride بكربون أتوان fat بكربون أتوان protein بكربون كسورس إنرجي بهار RBC شوي citrinium okay Glycolysis is for ATP, for energy. Hexose monophosphation is for an antioxidant. It's not for ATP generation. It's like an antioxidant pathway. I will tell you about this, okay, later on. So how the glycolysis has been controlled? We say hexokinase and glucokinase. Phosphorylation of glucose in important steps, okay? So makes glucose an ionic, as your colleague says. Increase reactivity of sugar. Make high energetic compound, but the main aim of hexo and glucose was to trap glucose inside the cell. Okay. What about PFK1? We said it's a key enzyme. It will be inhibited by citrate and ATP and will be stimulated by AMP. Hormonally in the liver will be insulin stimulate and glucagon inhibit PFK1. طبعا insulin و glucagon عكس يكترم. زور بين حالة كان كinsulin stimulate هرمونك كان بالعكس glucagon inhibit يكا. Okay? So it's a counter-regulatory hormone of the insulin. يعني راكب عكس insulin ويشكا glucagon. So if you have to guess which enzyme inhibit PFK1, for example, in the liver, say glucagon because insulin stimulated. Glucagon will inhibit it. So we said in the anaerobic glycolysis, what's the product of anaerobic glycolysis? Is what? Lactate, yes. So what will happen to this lactate, or what's the significance of lactate production? So we said glucose in glycolysis become pyruvate, and pyruvate will see if there is no oxygen or the cell have no mitochondria, we'll see this enzyme that's called lactate dehydrogenase will generate lactate or lactic acid, and this lactate will enter into circulation, start to rise. The lactate طبعا LDH أتواني إيش لسه lactate إيش كا إيش تلاقي لسه pyruvate إيش كاني بهدو لأنك أتواني دروسيكا أتواني pyruvate بجورد والlactate وأتواني بالعكس إيش lactate جورده back to the pyruvate. Okay? So LDH is reversible step يعني تلاقي بهدو سلكة أتواني إيش كان بس substrate to target وبالعكس إيش يعني pyruvate أتواني بجورد بالlactate lactate يجي back to the pyruvate. So lactate by LDH will go back to the pyruvate, and pyruvate enter into liver making glucose. This is a step called gluconeogenesis. I will tell you in the other lectures, don't worry about that. And also can generate a CO2 in the heart, okay? So this is how the lactate works. So what's lactic acidosis? What's lactic acidosis? Decrease? Why? Because of what? Increase, yes. So lactic acidosis means elevation of the level of the lactic acid. Why this will happen? If there is increased production, include when you do a strength exercise, shock, and congestive heart disease. Why in heavy exercise you develop lactic acidosis? زور باشا لبرويكا أكسجينيكا تو بريدينيا كي بو إكسرسايزكت هشتا بشي مسلكت ناقة. So بشي كي مسلكت جت سرشي anaerobic glycolysis. So in heavy exercise there will be increased production of lactic acid and lactic acidosis. What about shock and heart failure? Very good. So there will be decreased oxygenation to the cells. So whenever the cell lack oxygen, it will develop lactic acidosis because glycolysis will go into an anaerobic pathway. What about decreased utilization occur in the liver disease when liver cannot use lactic acid, or thiamine deficiency? We will talk about it later on. And during also alcohol metabolism, don't worry about these now. Okay, so these are my references for your lecture. Thank you very much.
Now, the blood normally contains a wide range of substances that can be used for diagnosis of a disease. These parameters or these metabolites in the blood may be a nutrient to the cell or maybe a waste product or byproduct of metabolism of the cells. However, if you go to the laboratory and do blood test, جاي أجيب مختبر فحصي للابكا شيء كان measurement waste product كان يا cell nutrient بوا أزانن كتو you have a specific type of disease. This important window, you can see the patient status. So by blood examination in the lab, you can measure waste product, like what? You can measure waste product like urea, which is the end product of protein metabolism. If someone have high level of urea, you say this patient have renal failure or renal disease. And also we have creatinine, which is also this is a waste product. What about the nutrient? جايباتو blood فحصكي nutrient كمان فحصكي. أزاني deficiency وهي يعني بعض النيوترينت هي بذبته like what? Glucose. If someone have very high level glucose, this patient have hyperglycemia and they may have diabetes. So هذا nutrient أتواني measurement كي لقى waste product بقول شيء كي diagnosis disease كي. زور شد همو زور بي زوري نخوش كان ببلاد examination أزاني توجي أم examination شيء waste يا nutrient يعني بقول هنا هيك بتحسي B12 فقط بيزاني vitamin B12 لويا يا شيء يعني vitamin D3. These are nutrients of the cell. أو كاتب عليه deficiency وهذا vitamin B12 يعني deficiency وهذا D3. This level, these parameters or waste product are in a constant level. What is the meaning of that? Example, creatinine in the blood have a narrow range. 0.6 to 1.1 is narrow range. This range remains constant. And there's a change that occurred in case of pathological and physiological situation. This narrow range, if there is a very high level of creatinine, means a disease. A bit, the level of this substance will be, there should be a constant level of it. But change occurred in case of physiological condition, like what? If someone have very large amount or large muscle mass, a male athletic have high level of creatinine. In case of pathological, the physiological, the pathological, there is a change. And after meal, if someone ingesting large amount of meat, urea urea. This is physiological condition. Glucose blood glucose these are physiological, يعني نخوشينية. ترى hypoglycemia مثلا blood glucose sugar drink soda blood glucose these are physiological. These there is changes in case of pathological, in case of disease. وكوباس ماركت glucose أجريك هالمعدل برز glucose أجري سرود دوصلوا. This is diagnosis of diabetes mellitus. Diabetes mellitus. In case of obesity, what do you expect? If someone have large amount of fat in the body tissue, what do you expect in the blood? Hyper? No, in the blood. Blood examination. Hyperlipidemia, very high level of triglyceride and cholesterol. In case of malnutrition, and there is certain enzyme deficiency, like what? Genetic disease. Genetic disease, they have enzyme deficiency. Like what? A disease that have deficiency in the enzymes. Just one example.
Anemia.
Which enzyme is deficient? I will give you an example, like G6PD deficiency. If you estimate the level of glucose G6PD enzyme, there is low activity of this enzyme. And what about the high level, high increase in the activity of the enzyme? لچكاتك الرواية هاي أمروخين كان ده أجل يكتب liver الروحيني هذا hepatic liver enzyme بس بيتوا the very destructionist يلا كان بيتوا هو أنزيم كان تتناول له بس بيتوا عادة لزوربي زوري disease there is increase in the enzyme activity while in genetic deficiency there is decrease in the enzyme activity like G6PD. So this nutrition and this nutrient and waste product may be changed in case of physiological and in case of pathological condition. Look to this. أم نمبرانا إيش داخلني أبو الامتحان بلاي لبالي كم بلان أمانة هي لبلا دا glucose. Amino acid is important. It's an important building block for what? Muscle. Hey, amino acid. Cholesterol, fatty acid, lactic acid. Lactic acid is increased in case of what? If there is anaerobic metabolism, in case of shock. If there is no oxygen to the tissue, lactic acid is a waste product. Again, shock there is no oxygen perfusion to the tissue, and lactic acid will increase. Urea and ammonia. Ammonia with manbochi خرافة. بس ما كده برو 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 بنا بنيوا. Ammonia is a toxic substance. If ammonia cannot be converted to urea, ammonia will increase. These are important waste product and nutrient inside the blood. هيك كيف فحصك هذا؟ على فحصك مختبري فحصك عام وبوكا. شيء بيتون nutrition كان nutrient يا waste product يا enzyme activity. أمشانا أكيد دواي سيرك أي بي الله توشوريك وبزا. شكرك وايا قلت له طبيعية جيجيرة طبيعية بتشي لبريك زي أمشانا وكلوا. nutrient and waste product لشو هاتوا لنيوتريشنوا تو خواتنا خوي glucose برزبيتوا or sometime inside the body there will be formation of these substances like amino acid and also this release of the store. Example liver glycogen تاعها ام glycogen شيء degradation of glucose و blood glucose normalize كاتو in case of short stage fasting. These nutrients are important for the production of energy, formation and building block, formation of the building block of the cell component, and also storage. And also there is interconversion of this substance to other. Example, amino acid will change to glucose, glucose change to fat. Which substance is important source of storage energy? Fat. What about glycogen? Glycogen is a source of. What is the difference between glycogen and fat? Glycogen. Glycogen in milligram, while fat in kilograms. هيك هالفوق همية بفات لا شيء 20 كيلو فات يعني. كذا سايزه كيف فرق هي؟ بالله فرق. كم؟ Synthesis of cell component. ما بس يوي جلوكوز مثلا يعني امينو اسيد بكاريه بو دروس كدني muscle lipid important for the formation of cell membrane. These nutrients there will be extensive interconversion inside the cells of this substance called what? من يكفي كم أي كابتوان.
هو فرق نية
Inside the cells, interconversion of these substances, example glucose to metabolic pathway. You have different metabolic pathway in the biochemistry. And also there's a metabolic map. Metabolic map here, یەک پەڵی گەورەیە ئەمە کاربۆهایدرێت میتابۆلیزم ئەمە لیپید ئەمە پرۆتین دوای هەموو سەیرەکە زانانی ئەمە مۆلیکوڵە لە بیدایەتەوە تا نهایەت ئەبێ بە چی کار ئەبێ بە CO2 H2O ATP یعنی دەرس میتابۆلیک ماپ خەریتەیەکی گەورەیە دوای سەیری هەمووی کەی most of the pathways, okay, all types of cells. The most important tissue for the metabolism is liver. Liver is really a metabolic factory. مشتقاً بزوري لليفر عروقيا. The most important source of energy is what? ATP. And also we have substances called reducing substances like NADPH. These are important source of electrons. This reaction occurs in the mitochondria inside the cells. In biochemistry, the location of the organelle are important. Location. جاري الله مثلا ATP لكوا production أبي بزاني لتي organelle كادو سبيا. cholesterol لتي شوني كي سيل كادو سبيا لcytoplasma. يعني the location inside the cells and the membrane are important in biochemistry. This diagram shows you the simple overview of metabolism. You see nutrient change to the building block molecule. There's catabolism of these large molecules to smaller molecules. And finally, they will give CO2, H2O, and NH3. These building blocks are important for the formation of cell component. What about this reducing power? What man reducing molecular? NADPH and also FADH. These are reducing molecules. These are vitamins. They are found inside the mitochondria. And energy produced inside the mitochondria. Energy is important for what? For the function of the cells. If someone lack ATP, what happened? Again, ATP block ATP بدوس نبه. Cell function باش نبه. Again, cell function باش نبه. إبتهويجي death of the cells, destruction of the cell membrane. That's to say ATP is very important for the maintenance of the cells. There's different types of pump inside the membrane of the cells. If there is no ATP, there is no function of the cells, and finally, these cells will be dead. This is simple overview of metabolism. Nutrient management. We have macronutrient and micronutrient. What are these macronutrients? Chief man here. No, بعام شيء معناه. We have protein, carbohydrate, lipid, protein, carbohydrate, fat. باشا إنت لما تقول هاتيك كان خوار أبي بزانين أما شيء lipid, carbohydrate, fat. This is very important. زور مهم بزاني شيء. أو شيء كي خيط الدم دوي خوي. شرنية خوار بالبيت. شرنية. استوايدي هاتوا بجدي. أزاي شونا؟ استوايدي هاتوا. بو نمونة كتاب بوينه. أنا highly processed food here. Highly processed food. مثلا شيبسي أخوي. تيستي زور خوشي هي. تيستي زور خوشي. بلا أو تيستا real food نية. real food نییە سەبستانسێکی تیا ئینهانسی تەیستەکەکە چۆن بڵێ وا لە دوو ئەکە هەستکەی کە تامی زۆر خۆشە ئەن ریاڵیتی this is not food یعنی خواردن نییە تۆی بەڵام چۆن بڵێ برینی تۆ نیورۆترانسمیتەرەکان وای لێکراوە کە بڵێ ئەمە خواردنێکی زۆر خۆشە زۆر زۆر بخۆی وەکو نانەوردە وایە کەچی خۆی زۆر زۆر خۆشە وانە these are not real food لەبەرەوە لێستا ئاگاتان لە خۆتان بێ بەس real food خۆن باشە Anabolic and catabolic reaction. This is simple and easy. In case of anabolism, what happened? These macronutrients, carbohydrates, fat, and protein, in case of anabolism, will change to polysaccharide, lipid, nucleic acid, and more complex substance. کەواتە تو ئەگەر پرۆتین خۆی مەسەلەن پرۆتین ئەو پرۆتینە هەبێ بەچی مەسڵ دروستەکە this anabolic کەواتە تو هەر شتێکە خوارد فودێکە خوارد بە زۆری anabolic بێ باشترە and also we have catabolic what about the catabolism destruction of the molecules to smaller one to produce ATP کەواتە ئەم ATP لێرەوە دروستە بێت بەکارهێنرێ بۆچی or anabolism. We have two types of reaction, anabolism and catabolism. And also we have two types of oxidation and reduction. This type of reaction mostly occur in the mitochondria. Oxidation is removal of hydrogen atom, while reduction is addition of hydrogen atom. Oxidation reduction mostly occurs in the mitochondria of the cells, site of the reaction inside mitochondria. In case of oxidation, there's removal of hydrogen. While reduction is addition of hydrogen ion. You see oxidation and reduction done by NADPH. This is oxidation in which there is removal of hydrogen ion, while the other side is reduction, addition of hydrogen ion. Of reaction inside the mitochondria in which there is production of NADPH, and this NADPH is important source of energy for the production of the fatty acid. This is called reducing powers, NADPH. This will change a substance, example pyruvic acid to lactic acid. ماذا تفهمون؟ خمر علاقة بهيدروجين كوية. Hydrolysis is a serious reaction inside the mitochondria that will produce certain amount of ATP. I hope you're reducing equivalent to NADPH, FADH amana, reducing equivalent to the mitochondria ATP P7. Bala. have two types of reaction inside that cell. One that produce energy or heat called exergonic reactions. Other that uses heat or energy or requiring energy called endergonic reaction. The change is called enthalpy. بس نعطينا لبركنا مو بشوم ليشتكي فيزيائية بس نعطينا we have two types of reaction. One need energy, other require energy. The difference is called enthalpy. Substance او indication işleyek. Onu glukoz beresbe, insulin işleyek. Urea beresbe de renal failure. Ammonia beresbe de urea cycle defect. Ama şey ki koyuyor. Fark.
أول شيء في الكربونات بيسكها ميزاني.
Değişnaka. مفهوم سؤالك عندنا بس. Jak tam nawie Zachman? يعني بتاني شيء كوروايا والله والله there is no shutdown, there is equilibrium. مو reaction على السيل هم equilibrium. يعني أولية هو عملية كيميائية بروا بشكل هم يلها وكشاع لإكسانية. We have ATP and ADP. I will be using that as an ATP as an. If there is large amount of ATP inside the cells, if there is large amount of ATP inside the cells, this will give a negative feedback for energy producing reactions. فهذا أجد ATP منظور به يكسري لنا mitochondria على stop making ATP. Sometimes this causes negative feedback. Well, if you have a large amount of ADP or AMP, this means there is a deficiency in the ATP. This will give a positive forward or positive feedback inhibition. يعني أوقات أبد هو يريكا ATP كم بيت على ATP دروس كأجد ATP زور بيه على كم يكره. This is very clear. هاي point مهمة بقوله. ATP signs of large amount of energy. ADP is a sign of deficiency of the energy. There is a signals inside the cells. These signals are important for the production of energy or inhibition of the energy production. ATP is a signal that there's a high energy signal. There's no need for production of more ATP. ADP and AMP are low signals, are low energy signals for production of more ATP. And also NADH NADPH, FADH, D2H are positive signs of energy. كواتا energy زورة. أو كي ATP أو energy زورة. أو كي كواتا reduced form are signals of high energy. Reduced form are signals of high energy. ماشي. أو كي بسور نسيو ملات سلايدك يا are positive signals for energy production. While the blue are the negative signals that tell the cells to produce more energy. كواتا شينك على زيادة energy تدوس كسورك على ميكا. يكم تشيبو أجل ATP هبت على energy دروس مكان السيل. كواتا أوي كرني السورة signy aware this cell have more energy. أوي كشينبو signy chia signy aware there is deficiency in the energy.
Back sugar
So it means that helps oxidation. Reduced factor help oxidation of ATP.
Which oxidation of ATP دروس هذا.
كمان يعني oxidation.
أول شيء يعني دعينا بس ما ندري chain reaction. We have compound that have high energy compound, أو energy كهتانة يستهيوا كان صالة كتير كمان بيتوا energy كهتانة energy دا عبازة energy زور مهمة وكشون بلاي طاقيتو. What are these molecules that have high level of energy inside the cells? The highest energy compound called phosphoryl pyruvate. Energy زور مهمة. هسه هيك كتير بقول workout energy نية. وانيا كواتش تك ورقريا pre-workout نيا شتك ورقريا بقول energy كابل. The highest energy compound is phosphoryl pyruvate. There's hydrolysis of this substance. And after hydrolysis, it will produce large amount of ATP. Second one, 1,3-biphosphogystrate. It has more energy. Creatine phosphate change to creatine plus phosphate will yield large amount of energy. However, creatine chia can be used. Creatine what she bakaya? Producing large amount of ATP. This is important for the athlete. Kawata, as I told you, Bodukani, Kawani Kashtika. Nutrition and absorption. They have creatine. Creatine for what? For increased energy. Basha. And ATP also produce energy. So ATP the highest energy compound is phosphoryl pyruvate. All the very PowerPoint can you We have this reaction, say creatine, you should differentiate between creatine and creatinine. Creatine is a waste product. ويجي يقول لي توابو حضرتك هو ما هو وقت كويس دوا دانش من توابو Creatine is a source of energy creatine plus ATP will convert to creatine phosphate and this plus ADP by the means of creatine kinase. And creatine kinase will change to a waste product called what? Creatinine. Creatinine is a waste product. Not creatine. يعني كواتا أويتا توي تريبو وجيمو workout ناي creatine. Creatinine is a waste product. Waste product. Waste product لكيو إيدا دروا؟
Addition.
Creatine لكيو excretion أبيا؟ Creatinine لرجع يوريا وإيدا دروا. If somebody have disease in the kidney, renal failure, creatinine will be decrease or increase? Increase in the blood. And the blood.
The urine is increasing.
And the urine may be decreased, but the blood is more important. That's to say, creatinine is a waste product, can be used for detection of renal failure. Creatine and creatine phosphate, there is change to a substance called creatinine. Creatinine is a waste product, and creatinine have no function in the blood. Only waste should be excreted in the urine. The rate of production of creatinine is constant. The rate of production of creatinine constant in the blood. كواتا أولي دقيقة بيجي كرياتينين يكبر ودواي ساعات كتير بيجي بدود بيه بس هاد constant. والله أجر برز جوا معناه شيء renal failure because there is no excretion in the urine. Because creatinine is mainly formed in the muscle, if someone have large amount of muscle mass, اگر ایچه ماسوتی زوره کریاتینی چیه؟ زوره بده برای که مصل زوره production زوره لسیلا. And what are the causes of low creatinine? نه اگر ایچه مصلی کم بیده چه حالت که مصلی کمه؟ elderly female يا حالتي تعيش
Elderly male.
هاش كسي ما أتروفيها بيه بلان female لبرو مصلي كم ترى أتروفيها كي زعترا كم بيه بلان بيت جدا ما يلو female أوانا كوزي كده
Malnutrition.
pathological if someone have amputated limb جاريوه كي قاتي نية قاتي كي براوتو سيركي كرياتيني زور لويا نازانی بۆچی کریاتنی وایە که بانگێکی دەژوورەوەڵی بۆچی وایە سەرەکی یا قاچی بڕاوەتەوە یا دەستی بڕاوەتەوە لە بەرەوەی مەسڵی کەمە هەتا مەسڵ کەم بێت کریاتنین کەمە. ئەمەشمان باس کرد high creatinine in the blood and low creatinine in the urine indicate what? Kidney failure. ئێوە ئەمڕۆ بەس باسی کریاتینمان کرد بەڵام زۆر سەبستانسی تر هەیە ئەینەن کریاتین. Example uric acid. High uric acid in the blood, low uric acid in the urine means what? Renal failure. باسی چیە گەنێمە؟ فلتەرێک هەیە، فلتەرەکە کدنیە. ئەگەر لە بڵەدا بەرز بێت، شتەکان لە یورینا کەم بێت، ئەوە renal failure. سوالی هەیە دەربارەی کریاتینین؟ ئەگەر چی کەم بێت؟ اگر مصل كم بيه طبعا اگر مصل ماسي كم بيه بلان او كم measurement and waste product یک دفعه جوت عن لبه كاتان زور ناكر للسيلات ده blood cell plasma or serum تيتيا ده blood nutrient تيتيا لقل waste product برزونزم كرنوي ام nutrient waste انا لشي الرأي لباثولوجيكال لفسيولوجيكال الرأي. What is the highest energy compound? Phosphoryl pyruvate. ATP is a source of energy. Creatinine is a waste product of creatine, which is the formation of creatinine is in a constant rate. If someone have high level of creatine and low in the blood and low level in the urine, this means renal failure. However, blood is a window for the patient status. يعني تقول the blood organism يعني بتشي هنا وشي هنا بتشي بمجرمنت يم أنا لايك أنا blood amino acid glucose amino acid urea creatinine and you have different types of analyte in the blood, each related to specific type of disease. كسؤالي هنا أو بس لازم بايو انرجيتيك وانا زور بيشوي نوع كل شيء اللي تبغى ترى فيزيكس نوت سو امبورتانت كلينيكال بوينت يعني زور ولا شيء دكتور زور مهم رقم ليفل ما رقم لهيك اكزامينيشن كان رقم نهائي أو إذا بومباس السؤال هنا